All right, so hello everybody. So I'm Clément, a PhD student, and I'm going to talk to you about the GxE prediction in phenomic selection using chemometric decomposition of NIRS data. So first, let me clarify a little bit what all of these words mean. So NIRS data stands for near-infrared spectroscopy. So it's just so NIRS data is just infrared spectra of parts of the plant. It can be grains or leaves or stems. Phenomic selection is when you try to predict your breeding values out of the NIRS data. So it's just like genomic prediction, but you replace the marker matrix with the near-infrared spectral matrix. And chemometrics is about extracting information from chemical systems by data-driven means. So data here is often near-infrared spectroscopy. And so chemometrics is a field of science that have developed a lot of methods to use and analyze near-infrared spectroscopy. The main difference between these two things are that in phenomic prediction, we try to predict the value of a genotype, whereas in chemometrics, we try to measure samples. And so then why would we need a chemometric decomposition of our NIRS data? So if you look at the classic GBLOP model that we use in phenomic selection, we compute the genetic similarity matrix as such. So we use S, the spectral matrix, and we compute the row similarity between individuals. But our spectra are phenotypes by themselves. And so we know from the quantitative genetics framework that we can decompose the spectra into a genetic part, an environmental part, and a GxE part. And so the hypothesis that we wanted to try here is that maybe using only the genetic part signal of the spectra is better to estimate a genetic kinship matrix than using the whole information in the spectra. So that's why we need a decomposition of the spectra. But like the most straightforward way to do so would be to take every wavelength of the spectra, compute such a model, and then assemble the estimates of all wavelengths to get the spectra. But we know that there is a high multicollinearity between wavelengths, and so multivariate methods would be more suited. And it happens that chemometrics offers a lot of multivariate methods specially designed for spectra analysis. So how do we try all of this? So we had a data set with a rice diversity panel comprising 188 genotypes. They were grown in 4 environments with 2 replications. And we also have S&P data for this rice and 1 spectrum per individual, so 1 spectrum per genotype, per environment, per replication. And so the core of the method we tested was with this classic GBLOP prediction model. And so the methods were about pre-processing the spectra in order to better calculate those kinship matrices that I've called Kg for the genetic term and KGE for the GxE term. So what are those methods? First thing we tried was the reference method that already exists in the phenomic prediction framework. So from the raw spectra, in each environment, you can correct for the experimental design, and you end up with one matrix for each environment, and each matrix contains 1 spectrum per genotype. So here S1 is 1 spectrum per genotype in the first environment, and S2 for the second environment, etc., etc. And then from each of these matrices in each environment, you can compute 1 kinship matrix. And so for the genetic term, you just take all your kinship matrix in all your environments, you average them, and you get your kinship matrix for your G term. And for the GxE term, you just concatenate all those environmental specific kinship matrix into 1 big matrix as such, and then you compute the row similarity of this matrix to get the similarity for the GxE term. Then the decomposition methods that we try, so the idea is to take the raw spectra and make it pass through models where you have a genetic term, an environmental term, and the interaction between the two, and you also correct for all the experimental design things. And then you take, so the blue or the blobs, like the genetic estimations, and you just retrieve, so the genetic spectra, which has 1 spectrum per genotype, the environmental spectra, which has 1 spectrum per environment, and with the GxE estimate, the GxE spectra, which has 1 spectrum by genotype environment combination. And then it's very simple. You compute the genetic kinship matrix with the genetic spectra, and the GxE kinship matrix with the GxE spectra. And you can also use the environmental spectra as environmental covariates, but I won't talk about that in the presentation. And so such decomposition models were done by two ways, either with a mixed model on each wavelength, and then you assemble the results of each wavelength to retrieve your spectra, or with LIMPCA, which is from the Chemometrics framework, and I will detail later what is it. I also tried another method from the Chemometrics framework, which is the orthogonalization, which is basically a way of extracting the variation that you do want out of your matrix. And so then, same, we take the orthogonalized spectra to compute the kinship matrix. So what's important here is that by comparing the reference methods with all the other methods, we compare decomposition versus no decomposition of the spectra. And by comparing the mixed model decomposition with chemometric decomposition, well, we compare classical decomposition versus the multivariate chemometric decomposition. So now I will detail what the LIMPCA and orthogonalization methods are. So first, the LIMPCA. So you take your raw spectra, the first thing you do is a PCA, which is just here to compress the data and then work on a few independent variables instead of working on 1,000 dependent variables. And then on each component, so on the scores of the PCA, you perform the decomposition model. And then, so you get the blues of or the blobs of the G part, and you just retrieve the genetic parts of the components, same for the environmental parts, and same for the GxE parts. And then on each one of these matrices, you perform a second PCA, which is here really to extract the signal out of these matrices. And you kind of go from the genetic parts of the components to the components of the genetic signal in your spectra. And then you can back transform all of these thanks to the loadings of the PCA into a spectrum. And so it can be quite confusing to have two PCAs in a row. So the first one is really here to handle the multicollinearity and work with a few independent variables, while the second one is really here to extract the signal out of the spectra. So that's it for the LIMPCA. Now the orthogonalization method. So you still go from your raw spectra, and you have to calculate a matrix of undesired variation. So that's the funky part, because there's no general way of doing this. But for example, it can be achieved quite simply. Imagine you have phenotypes of two genotypes, so G1 and G2, and you want the variation that you don't want is all the variation that is not due to genotypic differences. So to remove the genotype differences in your matrix, you can just center every data point on its genotypic average. And so you just remove the genotypic effects out of your data. And so this would be your matrix of undesired variation, because all the variation that's left here is not due to the genotypic difference. So that's what I did with my spectrum. And then on this matrix of undesired variation, you compute a PCA, and you get the loadings of this PCA, which represents the subspace of the undesired variation. And then you take your raw spectra, and you project it onto a space that is orthogonal to your loadings, and you get the spectra without the undesired variation. And if you want to put it mathematically, if X is the raw spectra and L are the loadings of the undesired variation, the orthogonalized spectra are X minus the projection on X of X onto the loadings. So now we'll get into the results. So here on the y-axis, you have the predictive abilities. On the x-axis, you have different traits that we studied, and they are ranked according to the importance of the GxE into these traits. So here, JW, grain width, has GxE is not very important for these traits. And here, for example, grain yield, GxE is very important for this trait. And so what we can observe is that the green line here is the reference model, and it really outperforms all the other models. The purple line is the mixed model decomposition, and it also outperforms the yellow and blue line that represents the chemometrics decomposition. There are two panels. This one is when we try to predict already observed genotypes in new environments, and this one is when we try to predict new genotypes in new environments. And we can see that in this scenario, the reference methods are outperformed by the decomposition methods, but still it seems that the mixed model decompositions still do better than the chemometrics decomposition. And this is all true only for the traits with a low GxE impact. So yeah, that's not really the result that we wished for and that we expected. So yeah, I guess it's too bad for my PhD. But anyway, we can try to understand what happens here and why is it so. And so we can first focus on the reference model. So here, it's the same plot as the previous slide, but it's a box plot, and there's only the reference methods. Red boxes, reference methods without GxE term in the prediction model, and blue boxes, reference method with the GxE term in the model. And you can see that there's no difference whether you incorporate or not GxE term in the model. So maybe our data wasn't really suited to study the GxE interaction and the GxE prediction. Next thing I want to talk about is the comparison between the decomposition and the reference methods. So in the decomposition methods, you take your spectra, you extract the genetic signal out of the spectra, and then you compute the kinship matrix with that signal. But the genetic signal of the spectra really is just the mean spectra of genotypes across environments. In the reference methods, you take your spectra, you compute one kinship matrix per environment, and then you compute your genetic kinship matrix by averaging all the environment-specific kinship matrix. So there's this same idea of using the environmental repetitions to estimate more precisely the genetic part of the signal. And so as these reference methods were really outperformed the decomposition methods in the results, maybe it's better to try to refine the kinship matrices once they are computed than trying to refine the spectra before computing the genetic matrices. Next thing and last point of discussion is that in the chemometrics method, they are PCA based, and at some point, there's always a step where you have to choose a number of components. And what I did here is choose the first two components with an eigenvalue superior to 1, because that's a classical way of choosing components. But if you look at your spectra here, the things that has the most variation in spectra are those differences, this kind of offset between spectras. And so this is the variation that you're going to retrieve in your first components. But this variation is typically due to the physics of the sample and not to the chemistry of the sample. And so by choosing the first three components, maybe I just extracted the physics differences between the spectra and not the chemistry of my samples, which may be bad for retrieving genetic signals, but may be good for like grain morphological traits, because grain morphological traits really define the physics of the sample. So, sorry, it's still work in progress. Analysis will be reconducted with suitable component selection methods. I'm also currently trying a PLS-based decomposition of the spectra and a GxE-based prediction models. And we'll also try those data on those methods on new data sets to see if like the results are due to the data sets or to the models that I've tried to compute. I still have a little bit of time. One thing that I forgot to put in my presentation is that Phenomic's prediction have been around since 2018, and it has proven to work in a variety of cases, but we still don't really know what part of the signal, what information in the spectra is responsible for the prediction, and we still don't really know so which part of the spectra we should try to extract in order to perform better prediction. And so these results may also tell us that the genetic signal of the spectra may not be what we should be looking for in order to increase prediction methods. So I'm done for my presentation. I'd like to thank my supervisor and the two chemometrics experts that helped me through this analysis. Thanks for your attention.